Hello, everybody, and welcome to webinar on the use of hormone therapies to induce spawning, spermiation, and ovulation in amphibians. I'm Dr. Aimee Silla, Research Fellow and co-head of the Evolution and Assisted Reproduction Laboratory at the University of Wollongong in Australia. And today I will provide a background and a framework for the application of hormone therapies to amphibians, and will present some anurin, frog and toad case studies. Following on from my presentation, you will hear from Dr. Ruth Marset-Greaves, who will provide an overview of hormone therapies for salamanders and will provide some practical demonstrations for you. So let's begin with a brief history of the use of hormone therapies in amphibians. The administration of hormones to induce gamete release was first achieved approximately 100 years ago. So this is not new technology, though protocols have advanced considerably over the years. Dating back to the early 1900s, formative research demonstrated a link between the pituitary gland and reproduction in amphibians through experiments involving the extraction of the pituitary and the administration of macerated pituitary extracts. This also led to the development of protocols using injection of macerated pituitary extracts that were taken from donor animals in order to stimulate gamete release in amphibians. Protocols that were used extensively throughout the 1920s and 30s onwards to facilitate research into developmental embryology. In 1930, Lancelot Hogben developed the Hogben test for human pregnancy diagnosis, whereby female South African clawed frogs, shown here, Xenopus laevis, would spontaneously release eggs within 8 to 12 hours after the administration of a single injection of urine from a pregnant woman. Later, it was discovered that male Argentine toads and northern leopard frogs could similarly be induced to release sperm in response to the injection of pregnancy urine, but within a shorter time frame and with greater reliability than female frogs. This frog test for human pregnancy diagnosis was used right up until the 1960s. And though they didn't know it at the time, the active constituent of human pregnancy urine that induced gamete release in frogs and toads was the hormone HCG. And we're going to talk more about HCG in a moment. The ability of laboratories to produce large numbers of amphibian eggs on demand from a small number of model species, including Xenopus laevis and Rana pipiens, really paved the way for a series of Nobel Prize winning scientific discoveries, including cloning, messenger RNA translation, and gene expression. Notably, the recent discovery that mature cells can be reprogrammed to become pluripotent has really revolutionized contemporary stem cell research and expanded our understanding of epigenesis. But despite this long and fascinating history of the use of amphibian gametes that are obtained via hormone therapies, the administration of hormones to induce gamete release in threatened amphibian species has really only gained momentum in the past 2 decades. But the number of threatened species within conservation breeding programs, or CBPs, to benefit from hormone therapies does continue to rise. There are three main reasons to incorporate hormone therapies into amphibian conservation breeding programs. The first is to enhance both the propagation and genetic management of species that are exhibiting reproductive failure in captivity. The second is to improve the genetic management of species that do breed successfully in captivity through environmental stimulation. And the third is to improve the reproductive health of individuals. So, for example, if females within a breeding colony remain gravid and fail to mate, then hormone therapies can be used to induce egg release so as to avoid egg retention and the associated health issues that may develop as a consequence, such as ovarian cysts. Before we go on to discuss current hormone therapies in more detail, I want to emphasize the importance of conditioning captive amphibians for reproduction prior to the application of hormone therapies. While hormone therapies can be used to control the reproductive physiology of amphibians in captivity, we advocate a more holistic approach whereby naturalistic, species specific environmental conditions, including climate cycling and adequate nutrition, are provided in order to assist in preparing and conditioning animals for reproduction prior to the use of hormone therapies. By adopting this more holistic approach, the quality of gametes and reproductive outcomes will be maximized, which will improve the efficacy of reproductive technologies and, in turn, the sustainability of amphibian CBPs. The provision of environmental conditions won't be discussed today, but I will direct you to a recent review article listed here, as well as a book chapter that will be published soon that provides an excellent practical guide for further reading on this topic. So, we can use hormone therapies to induce amplexus and natural spawning in male female pairs or groups of male and female amphibians. We can induce spermiation in isolated males, and the sperm collected can then be biobanked, chilled for short term cold storage, or it can be used fresh for artificial fertilizations, AF, also known as in vitro fertilization, IVF. And we can use hormone therapies to induce ovulation in isolated females. Of note, oocytes or unfertilized eggs collected from amphibians are too large to successfully cryopreserve, and fertilization capacity of oocytes does decline quite rapidly in cold storage. So it's best to collect oocytes and then fertilize them immediately. The process of conducting artificial fertilizations is going to be covered later in this webinar series. So now let's talk about hormones. Synthetic gonadotropin releasing hormone agonist, or GnRHA, also known as luteinizing hormone releasing hormone agonist, or LHRHA, and purified human chorionic gonadotropin HCG are by far the two most commonly employed exogenous hormones used to promote spawning in pairs of amphibians or to stimulate spermiation in isolated males and ovulation in isolated females for gamete collection. And these two hormones are widely commercially available. If we take a look at the HPG axis, you'll be able to visualize how the administration of each of these two hormones manipulates the neuroendocrine pathways that control reproduction in amphibians. First, let's have a look at the administration of synthetic GnRHA. GnRHA is structurally similar to natural GnRH molecules, and it acts by binding to receptors on the anterior pituitary to stimulate the synthesis and release of both luteinizing hormone LH and follicle stimulating hormone FSH, which in turn stimulates gonadal activity. HCG, on the other hand, is an LH agonist, attempting to mimic the LH surge and bind to LH receptors that are on the gonads. If you were in the previous webinar on reproductive endocrinology, you will no doubt have heard about the role of dopamine as an inhibitor of the release of LH from the pituitary. As a result, there has been recent interest in the combined administration of GnRHA with a dopamine antagonist, such as metoclopramide, to block the inhibitory role of dopamine and to enhance the LH surge. You can read more about the role of dopamine and the role of dopamine antagonists in the following reviews listed here. There does appear to be mixed results as to whether there is a benefit of the combined administration of GnRHA with a dopamine antagonist to induce spawning or spermiation, and so there is a current need for further research in this area. So now that we understand a bit about how the main exogenous hormones manipulate the endocrine system, where do we start in developing hormone therapies for a novel species? Well, successful protocols have been refined for a diversity of species of amphibians, and looking at established protocols is an excellent starting point. At the very end of my presentation, before I hand you over to Ruth, you'll find a list of recent review articles for further reading. There are several reviews on the topic, but the small number of them that I've listed have been selected because they contain comprehensive tables that detail established hormone therapies. Importantly, protocols are species specific, and it is essential that hormone therapies are refined for each species to determine. One, the most effective hormone type to administer, i.e., GnRHA or HCG, or whether a hormone cocktail such as GnRHA in combination with a dopamine antagonist is the most effective. Two, the optimal dose to administer by establishing a dose response curve. And three, the timing of peak gamete release for your target species. Importantly, optimal doses required to induce gamete release and the peak time for collecting gametes post hormone administration have been shown to vary considerably among amphibian species, and failure to quantify these appropriately may lead to incorrect conclusions when comparing the effectiveness of protocols. So let's move on to a couple of case studies. The northern crabbery frog, Pseudophrony pangilii, and the burralong frog, Latoria burralongensis. The northern crabbery frog is a small terrestrial toadlet that is listed as critically endangered by state and federal governments in Australia. The northern crabbery frog has been bred successfully in captivity for a number of years using appropriate environmental provisioning. And you would have heard a little bit about this if you listened in to webinar 5. The captive populations of this species display strong mating bias, with less than a third of available males contributing to mating success annually. And additionally, there are a number of females each year that are clearly gravid, but they fail to breed. Natural populations of this species also exhibit mating bias. However, it appears that mating biases are exacerbated in captive populations. And this is concerning because over time, such captive mating biases may lead to a loss of genetic variation, as well as the viability of the offspring that are generated for reintroduction. So, as part of a research collaboration between the University of Wollongong and Taronga Conservation Society Australia, we wanted to employ hormone induced spawning protocols to assist with the genetic management of the captive breeding program for this species. Hormone therapies had never previously been used for the northern crabbery frog. So, where to start? Well, fortunately, I had previously conducted research inducing spermiation in several Australian frogs from the same family, the family Myobatrachidae, including the northern crabbery frog's sister species, the southern crabbery frog, Pseudophrony crabbery. For each of these species, the effectiveness of GnRHA and HCG had been directly compared, and you can read about those experiments in the 2 papers listed there. The results from these 2 studies clearly showed that GnRHA was more effective at inducing spermiation in each of the species from the family Myobatrachidae. And so we focused on employing GnRHA at various doses that were within the range of doses that had been previously employed for these species here. We administered a single injection per animal of either 0, 0.5, 1, or 2 micrograms per gram of body weight of GnRHA. Hormones were diluted in saline or simplified amphibian ringer SAR and injected subcutaneously into the dorsal lymph sac. In experiment 2, 0, 25, or 50 micrograms of GnRHA was diluted in distilled water in order to promote absorption and administered in a dropwise fashion to the ventral abdominal surface. Each individual male and female received a single hormone dose immediately prior to being paired together in terrariums containing a gravel layer as well as moist sphagnum moss to allow for a terrestrial nest site for oviposition. And there was one male female pair per small terrarium. Hormone therapy was very successful. In experiment one, the optimal dose of zero point 0.5 micrograms per gram GnRHA injected resulted in 100% of pairs spawning, compared to only 22% of pairs that were in the control group. And in experiment two, 77% of pairs receiving 25 micrograms GnRHA topically spawned, compared to 27% of the control animals. You can read about the full results in the research article just here. Overall, in excess of 800 embryos were produced through hormone therapy over the study period, and these offspring have all subsequently been released into natural sites in the northern Brindabella Ranges by Taronga's Michael McFadden and DPI's Threatened Species Officer, Dr. David Hunter, both pictured here. Importantly, the offspring released had greater genetic diversity because hormone therapy allowed a greater number of males and females to contribute their genes to the next generation, as opposed to a few selected males that received all the mating success and a series of choosy females failing to oviposit. So we were really pleased with this outcome. But the research didn't stop there. We also wanted to employ and develop hormone therapies in order to induce spermiation and ovulation in isolated males and females of both the northern and southern crabby frogs, so that sperm could be biobanked and so that we could develop protocols for artificial fertilizations. So earlier this year, during the species natural breeding season in 2021, I induced spermiation in males from both of these species by administering a subcutaneous injection of either 0.5, 1, or 2 micrograms per gram GnRHA with 13 males per hormone treatment. Importantly, when inducing spermiation, frogs need to be kept separate in individual holding tanks, and it's important to provide a moist substrate in order to keep frogs hydrated, and this will allow for repeat sperm collection. A male southern crabby frog is pictured just here in this container containing laboratory tissue that's been wetted with RO water. Post hormone administration, sperm samples are flushed from the cloaca with the expulsion of urine in a suspension that is known as spermic urine. For the species that I work with, I collect spermic urine using a glass microcapillary tube, which is pictured here. The capillary tube is fire polished so that the end is smooth and does not abrade or cut the skin of the cloaca. The tube is gently inserted 2 to 3 millimeters or so into the cloaca and is retracted until urination occurs and spermic urine fills the tube via capillary action. Spermic urine can also be collected using plastic tubes or catheters, or alternatively, many anurines actually expel urine as an anti-predatory response. So it is possible to hold the male over a petri dish or a tray and to attempt to collect urine that is expelled that way. So in this study, spermiation was successfully induced in both the northern and southern crabby frogs. The experiment will be repeated, however, to increase replication this coming season. So I won't tell you about the optimal dose or the peak collection period just yet. But I will say that spermiation responses were very good, and the experiment also allowed doctors Justine O'Brien and Rebecca Hobbs to cryopreserve several sperm samples from these two species, and they are now held in Taronga's Cryodiversity Bank. We also wanted to induce ovulation in female northern and southern crabby frogs. Inducing ovulation in isolated female anurins can be more difficult to achieve compared with their male conspecifics, and priming injections and or hormone cocktails may be required. Unfortunately, an earlier attempt at inducing ovulation in the female southern Corby frogs was suboptimal, with approximately 30% of females releasing oocytes. And this was a study that I conducted with Phil Byrne back in 2010. In 2011, I conducted an experiment investigating the effect of administering various low dose priming injections prior to a higher ovulatory dose in a common model species. This was the West Australian Gunther's toadlet. A priming dose of 0.4 micrograms per gram GnRHA, followed by an ovulatory dose of 2 micrograms per gram, approximately 24 hours later, resulted in 100% of females releasing eggs. And as you've just seen there, that was a video of eggs being collected from this species, Pseudophrony guntheri, using a process known as stripping. Stripping is where pressure is applied to the female's trunk, encouraging oocytes to move down the oviduct, which is a process that mimics natural inguinal amplexus. In this study, 100% of females were able to be stripped of eggs, and each clutch was able to be fertilized with an average fertilization success of 97%. In subsequent experiments, the optimal protocol that was identified for Pseudophrony guntheri was used for additional IVF experiments in female P. guntheri again. And the protocol was also applied to two related species, Pseudophrony bibroni and Pseudophrony coriaceae, all with excellent success. So, based on these results, we decided to apply the same protocol to a small number of female northern and southern Corby frogs earlier this year. And you can see a video here of oocytes that are being stripped from a female Corby frog. Overall, the results were promising, with 5 out of 5 southern and 4 out of 5 northern Corby frogs responding to hormone therapy. But additional trials and fertilizations are going to be attempted in early 2022. All right, so I'd like to briefly run through another case study, the Burralong frog, Latoria burralongensis. The Burralong frog is a medium-sized stream breeding species from Australia that is listed as endangered by state and federal governments. A captive colony was established at Taronga Zoo, Sydney in 2008, and in summer 2020, an emergency rescue mission was instigated by a team from the Department of Planning, Industry and Environment, DPI, Taronga Zoo and the Australian Museum in order to rescue individuals from rapidly drying streams that were impacted by severe drought conditions. Approximately 10 years ago, not long after the captive breeding colony had initially been established, I set out to develop spermiation protocols for this species. But at the time, hormone therapies to induce spermiation had not been attempted in any Australian species from the genus Latoria. Fortunately, thanks to the efforts of Michael McFadden's team at Taronga, there were a large number of males available for this research. And so a large scale project was conducted involving 135 male burralong frogs, testing the spermiation response of males administered 0, 0.5, 1, 2, 4, 8, or 16 micrograms per gram GnRHA, or 0, 2.5, 5, 10, 20, or 40 international units or IU per gram HCG. Note that individual frogs only received one hormone treatment. I do not cycle animals through hormone treatments due to the potential of previous treatments to influence subsequent gamete release responses. So, this study clearly identified HCG as the most effective hormone to stimulate spermiation in this species at an optimal dose of 40 IU per gram. This study also determined the peak collection period for the species, as I collected spermic urine every 2 hours for 24 hours straight, and showed that sperm concentration and sperm quality were optimized between 1 to 6 hours post hormone administration. This led to a series of experiments by my PhD student at the time, Lisa Keogh, optimizing short term refrigerated storage conditions for burrelong frogs, as well as optimizing sperm motility activation protocols for this species. Having established the optimal dose and peak collection period for the burrelong frog, this also allowed for targeted collections of sperm from this species for biobanking. So last year, myself and Dr. Natalie Calatayud collected numerous spermic urine samples from male burrelong frogs housed at Taronga Zoo, which were then cryopreserved by Doctors Justine O'Brien and Rebecca Hobbs, and they are now held in Taronga's Cryodiversity Bank. With recent funding from the Zoo and Aquarium Association's Wildlife Conservation Funds, an excellent team are currently working on developing further hormone therapies and biobanking protocols in order to biobank sperm samples from a diversity of threatened Australian amphibians. Hopefully, today's presentation has provided you all with a framework for developing hormone therapies for the species that you work with. And I hope that you too are able to celebrate successes in the near future. I will now wrap up by providing the list of further reading that I promised earlier. So here's a list of review articles on hormone therapies for amphibians. And I will finish by thanking my collaborators, funding bodies, and by acknowledging the traditional custodians of the lands on which the research presented here today was conducted. Thank you all so much for listening, and I will now pass you over to the talented salamander love doctor, Dr. Ruth Marset Greaves. Thank you so much, and over to you, Ruth.
Thank you for that wonderful introduction. I am Ruth Marset Greaves. I'm the director of the Honduras Amphibian Rescue and Conservation Center, and today I will be talking to you about hormone protocol development in the caudates. So I will be a little redundant here and talking about the goals of hormone protocol development, but I want to, kind of talk to you about my personal goals when I use hormone protocols. First and foremost, do like to try and obtain natural breeding when I'm working with salamanders. There's not a whole lot of captive breeding colonies for salamanders right now, and so a lot of times we are trying to first just get them to breed naturally. And saying just get them to breed naturally is a funny thing because, as many of you know, getting amphibians to breed in captivity naturally is very challenging. So a lot of times when I'm using hormone protocols, the thing I'm trying to do is to stimulate breeding behaviors, to kind of give the animals a little extra something to get them in the mood and get them to breed, quote unquote, naturally. Then, of course, there are the reasons of stimulating gamete release, like in vitro fertilization, artificial insemination, and cryopreservation. That's, what I did my PhD on, and that is incredibly important for sustaining genetics and preserving a lot of populations in captivity. That's not always my primary goal, but that, of course, is very important use of hormone protocols in caudates as well. So when I'm approaching developing a hormone protocol for caudates, I want to make sure I understand the hormone cycles of the animal. When I go through this whole talk, keep in mind, there's not been a lot of research yet in caudates. At the end, I'll cite my sources and they will be very limited. There are some older papers out there with some minimal other protocols that I don't mention here today, but the more modern protocols that are being developed, there's not a lot backing them as of yet. We know they're successful and they're great, but that is not to say that they are the best. The best might be yet to come, and we're still working on things. So this is still new territory, and keep that in mind when you're working with salamanders. But developing hormone protocols requires you to have at least an understanding of how hormone cycles are going to work in these animals. You may not know what their hormone cycle is, but you may be able to at least go into it knowing the natural history of the animal, what their hormone cycle might be. So there are three general hormone cycles that we are going to be seeing in caudates, and those are associated, dissociated, and constant. The associated one is the one that we're probably going to most commonly think of because it's very intuitive. In an associated breeding cycle or hormone cycle, the hormones are peaked during mating season, the gonads are at their most active, the testicles are making sperm, and the eggs are fully developing or in the process of developing oocytes. And so it's associated with mating, this cycle. While it's fairly intuitive, the trouble with this is if you are trying to stimulate breeding more than once in an animal's typical breeding season. So many of these animals only breed once a year, and if you're trying to get this animal to breed more than once a year, and you give it hormone right after the mating season, that can overstimulate the gonads. And if you overstimulate the gonads, they won't produce again for quite some time, sometimes years, depending on the species. So you have to be careful with how soon you give hormones to these animals. It may not be safe. I recommend for any of these cycles, not just associated, that you wait at least 6 months between hormone protocol offering for females, and then at least 4 weeks for males. So constant and continuous, it's pretty self-explanatory. The hormones are constantly peaked and are regularly ready. The animals are regularly ready to breed. Now, this is not very common as far as we know in salamanders. It possibly is available in boletogloscids, so the more tropical salamanders. It's not something we're sure of yet, but the temperate salamanders, this is not something we regularly see. But this is something, if you were to give hormones to these animals, again, they probably would be ready and they would respond to the hormones, but you wouldn't want to give hormone too frequently, or they may have overstimulated gonads. Dissociated hormone cycles are the ones that give people the most trouble because they're a little unintuitive and a little difficult to work with. So dissociated breeders have hormone peaks and high gonadal activity not during the mating season. So typically these are animals that their sex hormones peak and their gonads begin development right before or during brumation. Now, a challenge with these animals is if you're thinking, oh, I'm going to give this animal some exogenous hormones and that's going to produce mating. That's not what's going to happen. If you give exogenous hormones, this animal, its body is going to respond to it as if, oh, it's time for me to develop sperm and eggs. It's time for me to, go into my dissociated pattern, and that will prolong the time before it's able to breed again. So with those hormone cycles in mind, and hopefully that wasn't too confusing for you, if you have questions, please feel free to contact me. I'll talk about some male hormone protocols and how different ones can be applied to different cycles. So aside from thinking about the hormone cycles, you should think about with males the spermatogenesis cycles. So when and how is the sperm being produced? Salamanders respond very much to seasonal changes, as most amphibians do. Most of the salamanders especially that we'll be working with in captive breeding colonies, are going to be temperate. So a lot of times we'll be wanting to change temperatures, we'll be wanting to put them into hibernation, and if possible, rain on them and cause thunderstorms and fake snow melt and things like that. Now, if you're not doing all of that, you still want to take into consideration the seasonality and when you're giving these hormones and how it's affecting their body, and mimicking the season within their body, even if externally nothing is changing. So the spermatogenesis cycles are, basically, are they responding to external cues or are they not? So there's discontinuous and potentially or semi-continuous seasonal trigger-based breeding. So discontinuous is usually if they have one breeding season. Potentially continuous just means they may have more than one breeding season. Some plethodons do that. They breed a couple times a year. And then continuous is not very common in caudates. I don't actually know of any that have continuous spermatogenesis cycles, because if you recall, salamanders produce spermatophores, and that isn't something that's frequently just readily available all the time. As far as what hormone protocols are available to you, I will kind of point out today which ones I recommend for which types of breeders, which hormone cycles, and which seasonality. But male caudates respond to a lot of different hormone protocols. During my research, I tested 16 different protocols, and the animals responded to all of them very similarly. So you have options. I did see, slight differences here and there, but not enough to be significant. And so I'm recommending what, I've seen work well in a lot of species. And so that's why I go with it. But we are still learning a lot about salamanders. And so there may be things that we learn in the future that work much better. And yeah, as we all work together to learn about salamanders, we'll figure that all out together. But that's just as far as, sperm release. As far as, if we're just trying to stimulate natural behaviors, there are a lot of different options too. Historically, there are some older papers that talk about using arginine basitocin, or ABT, and prolactin, PRL, on males to get them to produce pheromones and behave as though it's breeding season. And then more recently, I used topical GnRH in plethodonids. That paper is in review and will be out soon. And that was very successful in producing breeding behaviors naturally, quote unquote, naturally. So this is my generic go-to protocol for male caudates. Seasonality and cyclicity is very important, and I recommend cycling all your animals, putting them through changes in the environment, brumating them, and doing light changes, but often you're not going to know what the cycle of an animal is. And I understand a lot of times people aren't able to do complete environmental conditioning. And this is the generic protocol I recommend when you don't know what a cycle is, and you need to apply a hormone protocol. So there's two alternatives. The one I recommend the most is using a small GnRH prime, so 0.025 microgram per gram, and then 24 hours later giving a small dose of GnRH of 0.1 microgram per gram. That tends to give you higher concentrations and just higher quality sperm, just the way the sperm develops in the caudates, that seems to work with their physiology. However, alternatively, if you don't have the resources or the time to do a prime, 0.5 microgram per gram works very well as well. So if you do happen to know or suspect at the very least that an animal you're working with is a dissociated breeder, these animals need to be given hormones before either a brumation period, or if you're unable to brumate your animals, at least a dormancy period, a period where they're not being bred, and they should not be given hormone during a breeding period. So I recommend giving 0.5 microgram per gram of GnRH prior to hibernation if it is a short hibernation, so if it's a month hibernation-ish. Alternatively, if you're giving them a longer hibernation, so at least 4 months or so, I recommend giving a small dose of GnRH prior to hibernation, so 0.025 microgram per gram, and then about midway through, so about a month and a half to 2 months through, give them 0.1 microgram per gram of GnRH. If you happen to know, or suspect you have an animal that is an associated breeder and is a, semi-continuous or at least a seasonally responsive spermatogenic animal, I recommend giving them a smaller dose of hormone, because a lot of times if you're conditioning them, they're ready to go. And so their testicles have already prepared the sperm, and while you probably won't be able just to milk sperm out of them or get spermatophores out of them naturally, if you need a little hormone, I recommend giving a small dose. So my recommended dose, if you are cycling your animals and they seem to be very healthy and probably developing their own sperm, is 0.1 microgram per gram GnRH. Topical application is a very viable option, and we're seeing success with it. There's a paper coming out soon on its use in plethodontids. It has also been used in ambistomatids. With plethodontids, it goes over the face and down the back. Their pheromone receptors are on their face and over the base of their tail, so that is the best place to put it. And then for ambistomatids, it could be used in the cloaca. That seems to have good effects. In these species, it's only been used to simulate natural breeding thus far. It has not been attempted to use for stripping eggs or getting spermatophores or sperm separately. But that's not to say it can't be done. It's just not been attempted yet. But the dose recommended for this is 25 microgram per gram GnRH topically. So the basics of sperm collection, how you actually get sperm once you give them these, is just a reminder that the sperm in caudates is a little bit different than in anurans, so they keep their sperm separate from their urine, even where in some of the more primitive caudates where the sperm and the urine collect together or can collect together in the cloaca, they're released at a separate time. So you shouldn't be getting spermic urine in these animals naturally, but if you are artificially stripping the sperm from them, you may get urine and sperm mixed together. That will probably decrease the quality of your sperm. The spermatophore is the natural sperm deposit of most salamanders. The three most primitive families still produce liquid external sperm, but these ones, the rest of them produce spermatophores, which is developed for internal fertilization. So an option, depending on what you need sperm for, is to inject hormones to males, allow them to deposit spermatophores in the environment, and then take those spermatophores for whatever your use is, whether that's in vitro fertilization, artificial insemination, or cryopreservation. You don't necessarily have to strip sperm from males. You can use spermatophores. However, stripping the sperm from males can be very useful, especially in vitro fertilization, because you don't have to break down this dense gel from around the spermatophore. So milking the salamanders, I have a video demonstration of what it looks like. It's a massage of the salamander. So what you're doing is you're massaging their sides, you're massaging those tubules that I showed in that diagram earlier, down and pushing the sperm out of their cloaca. Remember, most of these animals produce spermatophores, and so what you're really doing is you're getting the sperm before it becomes the spermatophore, or you're sometimes breaking down an actual spermatophore depending how long into the process they are. You'll know if you're breaking down a spermatophore because you'll get some gel in there, but more often than not, you're just kind of getting a pre-metaphor, and you're getting some very dense, thick sperm. So here is a video demonstrating an injection, if that is the route that you choose to to offer your hormones. I tend to give it subcutaneously over the back, especially in the bigger salamanders. I will sometimes give it subcutaneously on the side or IP if necessary, peritoneally if necessary. But subcutaneously has always seemed to work very well for me, and it seems much less stressful. Because of their big tails, when you flip them over on their back, they will stress out and flip around. So over the back seems much more successful. And then I played this video last time, but I do recommend for animals that are very pheromonally inclined or stimulated by pheromones to give a little pre-stroke before you milk them. They do enjoy stronger, but they do respond to being stroked if they have been given hormones. So I give the hormones first, and then after I wait usually about 30 minutes, I stroke them and then milk them or allow them to deposit spermatophores. And so this is a video of the milking. So you'll see I'm stroking down the sides, and then you'll see the milk come out. And in this one, I collect it with a pipette. So just massage down the side, and it's a very gentle massage. If they're ready to go, there shouldn't be much pressure to it. Just a little pressure at the base by the cloaca. And that's that milt or broken down spermatophore, however you want to look at it. And that is a lot of sperm. So that's how you collect sperm if you're going to use liquid sperm and not just let them drop spermatophores in the environment. So female caudate hormone protocol development. Again, seasonality is key. In these ones, in the females, it's a little less complicated, as dissociated breeding is not very common in female caudates. I don't know of one yet that we have found that has dissociated breeding. I base my protocols on how gravid a female is. I tend to use ultrasound for that, but you can, gauge a lot of times if you know the female well and you've been cycling her on just how big she is. Natural conditioning of females is critical for success, and that is, as Aimee was talking about, everything: nutrition, environment. It's everything. So females need to be very well conditioned for good success rates. Ultrasound is very much your friend. I know not everyone will have access to this, but if you can get access to ultrasound, it doesn't even have to be a high quality ultrasound. I started ultrasounding salamanders with a cattle prod, and you can still tell where they are in their cycle. It's just, generic. Do they have big eggs or little eggs? It's invaluable to knowing where an animal is in the cycle and how much hormone you should use. So that is, what my protocols are based on. I have here how I base it on ultrasound, and then the next slide I have it, if you don't have ultrasound, what you can base it off of. But if a female is low grade on ultrasound, and we'll talk about this more when we get to ultrasound and what that looks like and what that means, I give them a prime of HCG 8 days before I want to give them an ovulatory dose. 24 hours before the ovulatory dose, I give them a 2 IU per gram HCG prime, and then there's an ovulatory dose of 4 IU per gram HCG and 0.1 microgram per gram LHRH or GnRH. Sorry, I copied this from my dissertation where we said LHRH. And then mid-grade ultrasound, it's the same, except you take out the 8-day prime. You just give them the 24-hour prime and the ovulatory dose. And then high grade, where it looks like they are just ready to lay, they only get an ovulatory dose. So, if you don't have an ultrasound, this is, the general rule of thumb, and you're cycling these animals and just guessing at their gravidity. So if you can't determine the gravidity, but they're very well conditioned and healthy animals, I recommend that longer cycle with the 2 primes and the ovulatory dose. If the animal appears gravid, but you're not 100% sure, but you think they're gravid, I recommend the one prime and the ovulatory dose. And if you are just, that girl looks like she is ready to burst, just give her an ovulatory dose. So some observations, there are some species that need that 24-hour prime, that 2 IU per gram HCG, no matter how ready they are. It will vary species by species. Um, it's not, if you don't know and you end up giving them an ovulatory dose and they don't lay, there are ways to help them lay those eggs. I know some people use oxytocin. I have found progesterone to be very helpful. And if you ever get in a pinch and have issues with animals not laying eggs, please just contact me and we can work through that. So caudate egg collection, what that looks like, I think we've already seen. So I always recommend using ultrasound. Even if you've given them the ovulatory dose, it helps to know if they've started resorbing eggs or how they look on ultrasound. And we'll talk about that more later. And then collection, it's, like Aimee said, it's called egg stripping. It looks a little different in caudates than it does in anurans. It's just a very gentle pressure. And then, the eggs start to come out if they're ready. If the eggs don't start to come out, you just give them a little bit more time. They don't tend to come out all at once. This video shows a very small amount coming out. Typically more come out than this. Typically,
Gentle
you'll
pressure. Okay, so
get, at least 3 times this much. But yeah, they don't tend to lay hundreds at a time.
Kind of have the dish help you along.
But again, we're still learning, and hopefully you all can help us learn more about salamanders. And if you have any questions, please feel free to contact me. I'm always happy to talk about my experiences and what you're doing and what we've learned. And thank you very much for your time.